Hi everyone and welcome to our video on sex linkage. What we're going to be doing in this video is having a look at specification reference 6.1.2b part 1, genetic diagrams to show patterns of inheritance including sex linkage, and 6.1.2b part 2, the use of phenotypic ratios to identify sex linkage to include explanations of linkage as well. So, first thing, if we're talking about sex linkage, we need a quick reminder on what these sex chromosomes actually are. So we're going to focus on humans predominantly in this video, but bear in mind that when it actually comes to your exam, don't be surprised if you get sex linkage in something completely random that you don't know about, okay? Don't be thrown by it. I know I've seen questions in the past on, I think it's pigeons is a favourite. Now, they won't necessarily have the same sex chromosomes that we do in humans, but all the information you need is in the question. So the principles of what we are doing in this video will apply. It's just read the question to make sure you get your notation for the chromosomes correct in whatever random creature you may encounter on your exam. So, back to humans then. In humans, we've got 23 pairs of chromosomes that make up our actual genome. Now, what we find is the 23rd pair are the sex chromosomes. Now, depending on if we're talking males or females, we have different sex chromosomes. So in males, it would be X and Y, and in females, 2 X's. Please be careful when we're writing this. Don't just write the sex chromosomes in male is just a Y, because you've forgotten half the chromosomes that exist as a sex chromosome. Just like you can't just write X on its own for females, you have to have the 2 X's. So make sure XY males, XX in females. Now, when we think about homologous chromosomes, then generally what we're saying there is that these two chromosomes match up. And when you look at those karyotypes, the pictures of the chromosomes, they kind of look the same, with the exception of this 23rd pair, the X and Y. Now, they're not fully homologous. In fact, we only have a very small part of these chromosomes that will pair up before meiosis takes place. And you can see the bits that pair up in the little diagram in the bottom left there. It's those little red bits on the tips, basically. Those are going to match up together. So on the X and the Y, they will match up so that we get them lining up in the correct orientation for meiosis to take place. But that's it. The rest of it's a little bit different. When we're then talking about these other 22 chromosomes in humans, they are referred to as the autosomes. Now, each autosomal chromosome pair is what we term homologous. So that means that they match in length and they contain the same genes at the same loci. So if we have two chromosome 22s, they will be around the same length. They will have the same genes at the same position on those chromosome structures as well. Now, if we think about the X chromosome, then it's actually got over 1,000 genes that determine characteristics or metabolic functions not linked to sex determination. Now, what we find is, because the Y chromosome basically is a bit shorter than the X, then it frequently doesn't have a partner for those genes on the X. So those 1,000 genes have something on the X chromosome, but nothing on the Y chromosome. So what this means as far as our two potential individuals, one male, one female, a female would have 2 genes for that condition. Because remember, a female has 2 X chromosomes. So if we're looking at the bit that say, let's just put a little mark up here. So if our gene is up here, then we've got 2 of them, one on each of the X chromosomes. In the males, obviously they've got an X and a Y. They're missing basically a section of that genetic material. These genes don't have that partner. So what we find is that we may have our little gene down here on the X, but there's nothing on the Y. So that means that a male will only have 1 gene in that position, which means that males are what we term functionally haploid, or we can call them hemizygous, So hemi there means half basically. So we can term them functionally haploid or hemizygous for these X-linked genes. These genes that occur on the bit of the X chromosome that is not present with a complementary gene on the Y. Perhaps it's easiest to have a little think about an example. So we're going to have a little look at haemophilia A. Now, haemophilia is all to do with your body's inability to clot its blood properly. So what we find is in haemophilia A, then this is all due to this non-functioning factor 8. Now, factor 8 is one of these clotting factors that exists in the body to obviously go through that whole very nice enzyme cascade of blood clotting that we've looked at previously. And it's coded for by a gene on that non-homologous region of the X chromosome. So what this means is that for a female to have haemophilia A, she would have to inherit 2 faulty alleles. Because she's obviously got the 2 X chromosomes, she would need the faulty allele to be on both of them to have this condition. If she only has a single faulty allele, she won't show the symptoms herself, but she will be a carrier of the condition. So that means that when she has children, they may then get this condition. If we then think about the situation with a male, then obviously they've got X and Y as their chromosomes that they're inheriting here, because this is obviously on the X part, the non-homologous region of the X chromosome there. All they need is 1 faulty allele and then they have haemophilia A. So quite clearly what we can see here is the odds are against the males in terms of haemophilia A. They are way more likely to develop the condition than women for the simple fact that they only have to have 1 faulty allele to get it, whereas a female has to have both faulty alleles to get it. Let's have a little look at a question that we could have. So work out the potential offspring genotypes and phenotypes for a mother who is a carrier and a normal father. So in the question, it is going to give you some element of notation, particularly if it's in an organism that is not humans, because you would have to obviously be told what the sex chromosomes are in order to be able to attempt this question. So what I've done in this little box here is just given you that key notation. So X is telling us it's the X chromosome, and if we've got a capital H, then that is a normal female. An X with a lowercase h is the faulty female, and Y, no superscript needed, because that gene doesn't exist. So Y just tells us that's the male chromosome. Okay, first thing to do then is let's get our little Punnett square set out. And again, you can draw these in the exam, that's absolutely fine. And remember our little standard way of doing these, males across the top, females down the side. So the mother is a carrier. If the mother is a carrier, that tells us that she has 1 normal and 1 faulty. So we need an X with a capital H and an X with a lowercase h. We have a normal father, which means that he's going to obviously have an X and a Y chromosome. And if he's normal, that means he must have the normal gene. So what we can see here then, same thing as always with our Punnett square, just fill out the boxes. Whatever is at the top goes in the 2 beneath it. Whatever is at the side goes in the boxes to the right. So we end up with this little setup. Then all we've got to do is identify the phenotypes. So first one is going to obviously be a female because we've got 2 X's and they're both capitals, so that is going to be a normal female. Top right, we've got an X with a capital H and a Y, so that one is going to be a normal male. Bottom left, we've got an X with a capital H and an X with a lowercase h, so this is going to be a female, but she's going to be a carrier because she's obviously got 1 of the faulty alleles. Won't show symptoms herself, could give it to offspring. And then in the bottom right, we've got a male because we've got the Y chromosome present, and because it's a lowercase h on the X there, then this means that they have the actual condition. So this is our haemophilia A male. So basically what we can see from this is that you have got a two, okay, so two of these potential offspring are going to be normal non-carriers. One is going to be a carrier, could only be female as a carrier, remember, and one is the male with the condition. We can also look at another example just to make sure we all understand a few little quirks, let's say, of nature, which is if we delve into the world of cats. Now, cats have one gene for coat colour that is sex-linked, and it's all to do with if they are orange-furred or not orange-furred, basically. So we've got orange fur or black fur as our options here. So C is standing for our coat colour, a capital O in the superscript position tells us it's orange fur, and a capital B is a black fur. Now, both of these alleles are codominant. And if they're codominant, remember that just means that they are both going to have an effect on that final phenotype. So if we end up with a cat that has both the orange and the black fur, then what we find is they show both orange and black fur in a pattern. And this is called tortoiseshell. For those of you that haven't seen a tortoiseshell, there's one example of a little tortoiseshell at the bottom, but basically they have little patches of orange and patches of black on it. Now, the key thing about this is that if we look at them, it's a case of they've got very much these little bits of fur that are orange and other bits that are black. Now, if obviously all of their cells have inherited the same DNA, then they will each have orange and black fur genes. So why are some orange and why are some black? Well, it just comes down to this idea of inactivating one of the X chromosomes. So basically determining which bit of fur is black and which bit of fur is orange just comes as the result of one of those X chromosomes being inactivated. And the reason this has to happen is that if we just think logically, then when we've got a male, which is obviously X and Y, then we've obviously got a region where there are no partner genes on the Y chromosome. If we've got the two X's, that means that entire region, all of those genes located in this region that doesn't exist on the Y, have basically an additional gene coming into play. So what we find is we have to inactivate one of them to avoid issues. So this is something that's going to happen very early in the embryonic development of these mammals, and it's a completely random process. Hence why when you look at those tortoiseshells, they've got these odd little patches of orange, little patches of black scattered all over. It's because at that very early stage of the embryonic development, one of those X chromosomes was inactivated. So if it had been the orange one that was inactivated, that would be a patch of black fur. If it had been the black one inactivated, then we get the patch of orange fur. And because it's completely random, that means it's not a case of you have a half black, half orange cat. It's just down to complete chance. As always, I hope you found this video useful and that you now understand how this sex linkage works. As always, I suggest that you subscribe to the channel so that you know when I next upload another video, and of course head on over to the website so that you can see the other useful resources that are going to help you in your A-level studies.